So our project, which I think really follows very well on Yang's project, is to describe the RNA dynamics that's underneath facial development. And this is a collaboration between myself and Ken Jones and Trevor Williams at University of Colorado, Denver Anschutz Medical Campus. So when we're talking about the developing face and its morphogenesis, we're primarily interested in the earlier events, 9.5 to 12.5. This is when the majority of the morphogenesis, the major morphogenesis that generates a recognizable face. And not to go into all the details of that because we've heard a lot about it and most of us know much about that. But really what I wanted to focus on today is this issue of the RNA dynamics. So we've been talking a lot about the genomic and genetic programs that underlie the development. We've been talking a lot about the morphology and the morphogenesis that we see. But we know, but tend to ignore the fact that there's a lot of steps between. Is there a pointer up here?
There is.
Yeah.
Push that green button.
There we go. Okay. Yes. Lots of steps in between transcription of the genome and the cellular behavior that's mediated by the proteins that are encoded. And dealing with the enhancers, the transcriptional apparatus, maturation of the transcripts, and then all the various different choices that happen about whether those transcripts are sequestered, degraded, or actively translated. And there's a fair amount of data now that shows that probably more than 50% of the variation in protein, or I should say, if you look at the levels of proteins, less than 50% of that can be actually predicted by the steady state levels of the mRNAs that we generally measure in our transcriptome studies. And so our contention is that it's really very important to understand not only the steady state levels of transcriptome, which is measured in most RNA-seq experiments, but also all the dynamic regulation of those mRNAs that take it to get making the proteins themselves. And so our project is to really do a whole series of different measurements to follow that RNA dynamics. And really, so the initial proposal was really quite broad, but as it was funded, we're now focusing on from the transcriptome and then what happens in its maturation, so the splicing and processing, and then post-transcriptional events leading to translation. And so we've got a series of different data that we're going to be doing from the RNA-seq, ribosomal profiling, small RNAs, and from these, what we really want to do is to derive for every gene or for every process a description of at what different levels are these processes regulated. How much is regulated by RNA turnover, how much is regulated at the level of enhancer or transcriptional rates, how much is regulated at the level of translational rates. And so that's the overall picture and objective. But beyond that, there was a second thing that we wanted to incorporate into this study, which has to do with tissue differences. So again, we've heard a lot of talks about the neural crest cells and the mesenchyme, which will constitute the bulk of what we see in the face. But it turns out that the ectoderm is a really extraordinarily different tissue with a completely different genetic program, and that the events that happen in the ectoderm and the communication between these two tissues is absolutely critical for correct facial patterning. Just an example over here on the right is an example from some microarray data that we had earlier on, showing that the ectoderm and mesenchyme gene expression patterns programs are just dramatically different. And here on the right, to show that if you manipulate gene expression in the ectoderm, you get profound effects on the facial morphology. And so this is just a reminder that what happens in the ectoderm is very important. And so we want to follow the RNA dynamics of both the ectoderm and the mesenchyme. And so to do so, the general approach would be to do microdissections of the 3 different facial prominences, the mandibular prominence, the maxillary prominence, and the frontonasal prominence. And then from that dissected prominences to actually peel off the ectoderm so that you can get ectoderm and mesenchyme from each of these different facial prominences, and then to use that tissue as input for the various different assays. And so now just what I want to talk about briefly. We've had about 6 months working on this project. We came a little bit late to the FaceBase too. And so what we're focusing on first is the transcriptome itself, the mRNA, RNA-seq, and the small RNAs. And I just want to tell you a little bit about the progress we've made on that so far. And so in terms of the transcriptome, the RNA-seq, we're starting with the E12.5 data because that's when the prominence is largest and it's easiest to get the tissue. And we've, in fact, now got pre-processed data for RNA-seq for E12.5 from the 3 facial prominences, so frontonasal prominence, maxillary prominence, and mandibular prominence, for the ectoderm, for the mesenchyme, and it's also got the nasal epithelium, which is the neural epithelium, which is tissue quite different. Triplicate samples for each of those, and just getting the data last night and today. But so the data we're doing 125 base paired ends sequencing. And we've got nice deep data, excellent QC in terms of reads passing QC. They're all mapping really very nicely. And so it seems to be high quality data that we've gotten. And in terms of the biological relevance of the data, if we just look at principal components on where they lie, we find that the nasal epithelium group's completely different from the ectoderm, which again, completely different from the mesenchyme. So what we would expect is biologically different tissues. And in more detail, if you look at the ectoderm, you see that the frontonasal and the maxillary and mandibular group each with themselves. And in the mesenchyme, similarly, the frontonasal groups with itself. The maxillary and mandibular mesenchyme is really quite similar. This is, again, reproducing what we saw with our initial microarray experiments, where at least at this age, the maxillary and mandibular mesenchyme are really quite similar. So what we're taking away from this is that everything's working very well. We're getting high quality data. And essentially now we have the pre-processed data that's available for upload, and now we're taking it on to do the analysis of the isoform and splicing. Pipelines under development, working with this pilot data. And then the second piece of that isoform and splicing analysis would be verification. And so we've been talking about different ways to do high throughput verification for alternative splicing. And there's no really great solutions to the problem. But Affymetrix has come out with a new microarray that's really specifically designed to look at splicing. And so we've done some pilot experiments using that as a high throughput verification. And it looks really very promising that we're seeing a lot of confirmation from what our pipeline is producing from the RNA-seq data versus what the Affy arrays are giving us in terms of alternative isoform uses. And so it looks like we have, again, this technology is all up and working and ready to move forward with the 11.5 and the 10.5 tissue sets. So we're happy with this. And the other thing that we're working towards, which is a little bit more thorny, is dealing with the small RNAs. Okay, so when I'm talking about small RNAs, I think I'm talking about something a little bit smaller than Axel Fizzle was talking about with his enhancer-derived RNAs. So microRNAs that we're all familiar with, about 22 nucleotides. PiRNAs are a little bit larger. And then the small nucleolar RNAs, Cajal body RNAs, and the spliceosomal snRNAs, all less than 150 nucleotides. So FaceBase 1 had a RNA-seq project looking at microRNAs, specifically gel-purified size selected in this very small range by RNA-seq. that was mostly in secondary palate, but they also had some earlier tissues. So I just want to say a couple words about these classes of RNAs. These are sort of, I think, underappreciated but emerging as important regulatory classes of RNAs. So these, the SNO RNAs were initially recognized or described as being involved in the covalent modifications of ribosomal transfer RNA, so O-methylation, pseudouridylation. And but it turns out that there is a large number of these, maybe about 500 in the genome. It's a little bit under discussion exactly how many there are. And many of these are not involved in the canonical functions of T transfer and ribosomal RNA modifications. And there's an emerging body of evidence that they're involved in all kinds of other RNA dynamics, alternative splice selection, RNA editing, RNA cleavage. And even those that are involved in the ribosomal RNA modifications, there's some suggestions that these might be physiologically varying between different cell types and different conditions and have effects on cell behaviors. Much of that literature is coming from the cancer field, but there's really an emerging view that this class of RNAs could really be quite important regulatory elements in terms of genetic programs and tissue behaviors. And so we wanted to, if possible, capture these classes of RNAs as well as the more well-studied microRNAs in our studies. This turns out to be somewhat challenging for the reason that the tRNAs and the 5S RNAs, similar in size, similar in structure, and really it's almost impossible to separate them by physical type of methods. And it's also fairly difficult to think about separating them with sequence-specific types of methods because they're also quite diverse, and there's lots of different ones. So it's not as simple as just getting rid of the larger ribosomal RNAs. So it's a little bit of a difficult problem how to deal with this technically. Let's skip past that. And so again, the technical challenge is the diversity of the size. You have to get rid of these really abundant ribosomal tRNAs to study them. In terms of, there's all kinds of 5' end modifications that make them awkward to deal with in terms of library preps. And then finally, many of these library preps are really quite complicated, so that you get to the point where it's very difficult to have consistency from sample to sample to be able to compare. And so what we started looking at when we really got into this was alternative technologies. And again, now looking at the old-fashioned microarray technology, which has a lot of advantages in this situation. Because the sequence is on the chip, you design what you put there, you can completely ignore. You don't have to purify away things you don't want to look at. It's already on the chip. Of course, the disadvantage of that is that you will only ever study what you already know. And so there's good and bad to that. But we ran some pilot studies with this microarray chip from AFI. It's got about 36,000 genes on that. Most of them are microRNAs, but it's also got a series of other these SNO RNAs that we're interested in. It does not have the SN RNAs, the spliceosomal RNAs, but it does have these SNO RNAs on it. And so we ran a pilot study with that to see if it would give us the kind of data that we would like to get. And so, again, starting with 12.5 maxillary process, mesenchyme and ectoderm, using very different amounts of input, putting them onto the chips, and then asking, what can we detect? How's the sensitivity? How's the specificity? Is it giving us statistically reproducible data that we can work with? And so in terms of the sensitivity, we tried 3 different levels of input. We tried 12.5 nanograms, 40 nanograms, 125, and looking across all the different ways of looking at the data. 12.5 nanograms wasn't enough in terms of either detection or signal to noise or principal components, but 40 nanograms was enough, so that we know we're within the linear range of the assay here. And incidentally, we were very pleased to see that, again, the mesenchymal and ectodermal samples you have wide variation in the principal components, as we would hope to see, because we expect they're quite different tissue types. And then going on, are we detecting the kinds of things that we expect to see? And so for this, I took the data from FaceBase 1 from the RNA-seq studies of E12.5 maxillary process, and processed that through, they detected about 1,500 different genes represented in their small RNAs, many of which were microRNAs, either from mouse or other species, and then some of these other SNO RNAs and SNRNAs. So this we took as sort of a gold standard for what you might expect to see in these samples, and we asked how close the chips were to reproducing that. And so in our chip results, we saw it's really apples to oranges to try and compare these things. I will throw this out there in deference to Christine and her comment about 80% of the effort of reusing data is in cleaning up the data. I would say it's closer to 90%. Trying to work with this data and align the naming of these genes across these two different platforms is just a nightmare. And I will also say going forward that for FaceBase, it's something that if we're going to be talking about these small RNAs at all, we have to be very, very, very careful about our naming conventions and to do it right the first time. Okay, so with all those caveats, yes, we found a large number of microRNAs, as you would expect from a microRNA chip, and also a fair number of the SNO RNAs. So good news and the bad news. Good news that we found a lot of these, but the bad news, and again, the good news is that there was a fairly good overlap between the microRNAs that were identified by the two platforms. And I say 72% of what was detected here was also detected here. That's really a lower limit because a lot of the differences have to do with naming conventions. And it's probably much closer to 90% of what's detected here is actually here, if it were, if we could align them properly. The bad news, though, is with these small, the SNO RNAs, is that only 20% of what was detected in the RNA-seq data was detected here by the microarrays. And so there's issues there. And I think the most important issue there is that actually the genes that are on the microarray are the human sequences, not the mouse sequences. And although these are highly conserved in the 80 to 90% range, it's maybe not good enough for what we're trying to do. Get past that. Okay, and so in looking at a little more detail at specific things that we expect to find, for instance, we know that this microRNA cluster, 1792, it's very important in palatal fusion, blah, 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 blah. And all these microRNAs were detected by the gene chip, but we didn't see any differences in mesenchyme versus ectoderm. Another microRNA that's very important in the face, 140, promoting chondrogenesis via PDGF-alpha and DNPT. We found that, and we found it enriched in the mesenchyme, where you would expect to find something important for chondrogenesis. So that was really nice. Another microRNA, 200B, it's known to promote epithelial morphology and suppress epithelial to mesenchymal transition. And we found both the 5' and the 3' forms of that highly enriched in the ectoderm. So again, what we would expect. And so not only are we finding the microRNAs that we expect, but we're also finding them in the tissues that we expect. There's another study from a few years ago looking at primary and secondary palate microRNAs from E12.5. And of the 68 that they detected, 67 of them we also detected. Some of them in the ectoderm, some of them in the mesenchyme, mostly not enriched in either. And then what I mentioned before, the RNA-seq data from FaceBase 1. And so overall, in terms of the microRNAs, we're seeing good sensitivity, good specificity on the chip. And but in terms of the SNO RNAs, it's really not working very well at all. And so going forward, there's a couple of solutions to that. So we've decided that RNA-seq is just not an appropriate technology for what we want to do if we want to capture these SNO RNAs. And so one question that I would be throwing out to the community is whether this is important or not to do. It's really exploratory. We don't know if they're going to turn out to be important or not. It's not really been very well studied in any system, but we'd love to be the first to do it. That being said, the microRNAs detected by the chip seems to be pretty good, and most of what's found in the gold standard study seems to be already on the chip. There's some sort of we could invent a software patch that might be able to extract some more of the SNO RNA information. But really, probably the best solution, now that we've got through our pilot, I think we want to develop a custom chip that would have the mouse sequences specifically. And as long as that's not cost prohibitive, that's probably the direction we want to be going. And then the last comment was about the naming of these small RNAs. The naming, particularly for the SNO RNAs and the snRNAs, it's not standardized. There's not a community-wide consensus on how these things should be named. And so I think we need to be very careful going forward in how we're going to deal with it, listing these on FaceBase. And so just now to wrap up, so the aims of our project was transcriptional dynamics in terms of the large RNAs and the small RNAs. And our pilot studies, this is running really well. We've got the 12.5 data for the large RNAs, and we're well on track to get the rest of that data ready to upload. In terms of the isoform and splicing analysis, the software pipeline, we're working that through, and we're really very happy that validation that we've come up with, not the high throughput PCR, but the splicing microarray from API looks like it's going to be an excellent solution to that problem. And this is for the future. And so that's our progress at this point. And then I just want to give a shout out to Helen, who's sitting in the back here. You want to raise your hand? Who's done all the microdissections, and she's really the heart and soul of this project, helped by Reen. And Ken, who's also back here, who's all the bioinformatics, data processing, database work, along with Phil O'Neill. And with that, I'll take questions. Rich.
Well, Joan, you made kind of a big deal about the problem with nomenclature, and I don't get it. You've got an absolute standard, which is the genome sequence and build that the sequence is key to, and you could easily embed a Rosetta Stone with whatever you're calling something with what the nucleotides are. That way, anyone in the future knows exactly what you're talking about.
If you could summarize the question.
Okay, so Rich doesn't get the question of why it's why the naming conventions are a nightmare for the small RNAs. And so this is an issue of secondary use of the data. So if I go in as a secondary user, what I want to be able to do is to simply use, for instance, a RefSeq gene name to call up the gene I'm interested in. But if there's not a consensus on what the gene name should be, it becomes very difficult to compare the microRNA data or the SNO RNA data in this data set with a data set that's in some cancer-based encode thing that's in some other data set.
That's not your problem. That's not a FaceBase problem. So you've got a means for absolute definition of what you're talking about.
So yes, I do have a means for absolute definition of what I'm talking about, which would be essentially the genome coordinates. But this is, I think, also something that as a FaceBase community we need to be thinking about, is to how to make our data as accessible as possible to outside users. Question?
Yeah, so I was a little bit surprised about the sort of short RNA comparison at the SNO RNA level. So given the proportion of reads in the RNA-seq data mapped to SNO RNAs and tRNAs, I assume they did a size selection in the RNA-seq experiment. And that makes me wonder whether your comparison with the microarray data is actually valid.
So the question was about the SNO RNA sequences in the RNA-seq data, and whether that's a valid comparison between their data set and the newer chip. And the answer is, it's sort of like a apples to oranges to grapes to zebras comparison. Because for that RNA-seq experiment, yes, they did do a size selection. And so the SNO RNA pieces they're seeing are going to be either fragments that are from some kind of physiological processing or degradation. And so the representation is not necessarily going to be the same. I will also say that we've seen a lot of differential expression of SNO RNAs in the microarray experiments that we've done previously, where we're actually using the larger RNA fractions as our probes. And so I do believe that there's a fair amount of differential expression of this RNA class. I'm just not really quite sure that there is a gold standard out there for what we should be expecting to see.
And as a related question, presumably you know the probe sequences on the array, right? So you can check whether they're actually matching genomic sequences.
Right.
Can you rephrase that?
So I mentioned that the problem with the SNO RNA sequences on the array is that the probes are designed for the human, not the mouse genes. And yes, we know what those sequences are, and we can go back and derive new software that would actually calculate the mouse-specific signals out of that. But that's something that I would rather let AFI develop than do myself.
So, my way of clarification, so you said that RNA-seq is not suitable for SNO RNA. Is it because there is no appropriate statistical or bioinformatics tools to see the differential nature?
So the question is why RNA-seq is not so great for the SNO RNA studies. And the answer to that is really that if you were to do RNA-seq on those, the vast majority of the reads that you recover would be the tRNAs and the 5S RNAs, because those are much more abundant than the SNO RNAs that we're interested in. And so you would have to sequence to ridiculous depth to get the what you need.
I'm a little surprised that it was so hard to clean up the data. Aren't the people that generate the data like down the hall from you? We talk about that in private.
Yeah, we'll talk about that in private.
Okay.
No, that's again, the platforms are different. So the data from David's lab was sent to Oregon to be processed, and that's where the data repository is. And I think that what Joan's trying to say, and that's what Rich said, sorry, everybody, is that the nomenclature they're using for the RNA-seq reads is totally different than the nomenclature they're using for the chip. And to try and put those back together is very, very difficult. You actually have to almost go back to the sequence themselves to try and figure it out. Again, my understanding from the stuff from David Cludier is that there are a lot of single nucleotide differences in their microRNAs, which are confounding their ability to map them and analyze them. So they're not sure whether they're being processed after being made, or whether they are just bad reads, or whether they are different places in the genome. So that's a problem again with the RNA-seq. It's a good problem and a bad problem for those microRNAs.
So on that, how old? I'm new to FaceBase. Sorry. How old is the RNA-seq data that you were working with, and is it really comparable to what's available today?
Couple of years.
How old is that RNA? I can tell you, it's 2012.
That's old. That's ancient.
So in other words, we shouldn't store anything on the hub.
But that's a valid point. Is that you were into perpetuity. Things